Welcome back to the Deep Dive. Today we are strapping in for a really high-speed, fact-heavy journey through the essential mechanics of life.
That's right.
We've got our hands on a stack of advanced research material, and it's like a comprehensive map for the entire biological universe. I mean, from the architecture inside your smallest cell.
All the way up to global systems.
Right, things like disease resistance and conservation. So our mission is pretty clear. We are here to pull out the most essential, the most surprising nuggets of knowledge.
The foundational rules of biology.
Exactly. We're going to hit three linked domains.
Yeah.
The inner world of the cell, the molecular blueprints that control everything, and then how these systems maintain stability against, real-world chaos.
And for those of you who appreciate efficiency, this really is the ultimate shortcut. Biology can feel overwhelmingly complex.
Oh, absolutely.
But once you identify the core mechanisms, they are just shockingly elegant. And we have to start small. I mean, really small. At the level of the cell's detailed structure, what we call its ultrastructure.
To see something that detailed, we know we can't use the standard light microscope. That only gets you so far.
Not even close.
To really map out the internal scaffolding and all that tiny machinery, you have to use electrons.
You need the electron microscope.
Yeah.
And here is, I think, the first major mind bender about this technology. When you look at an image from one of these, you're looking at the effect of a beam you cannot see.
Wait, what?
The electron beam itself is invisible to the human eye.
So I always picture this like a super high-powered light, but you're saying it's more like an X-ray. If the beam's invisible, how do they even capture the image?
Exactly like an X-ray. The image is formed when the electrons hit a fluorescent screen, which then, projects the structure. And this is also why the raw image, before any computer enhancement, is strictly black and white. Electrons just don't carry color information.
That immediately reframes every stunning cell photo I've ever seen. But, hang on, biological tissue is mostly water, right? It must be transparent to electrons. So how do they get the contrast to see all the tiny details?
That's where the heavy metal atoms come in.
Oh.
We add contrast by staining the specimens with heavy metals compounds with lead or uranium, for example. These metals physically stop the electrons from passing through.
Just like calcium in your bones stops X-rays.
Precisely. So where the heavy metal builds up on a dense structure, it appears black in the final image, which means all those vibrant, colorful pictures you associate with cell biology are actually
fake.
Well, false color images. They're added later by a computer to make them easier to interpret.
It's a beautifully complex workaround to see something so small. Okay, let's unpack the cell itself. Let's talk energy production. You've got the two main factories, mitochondria for respiration and chloroplasts for photosynthesis.
Right.
But what's fascinating is how similar they are. It's almost like they were built on the same ancient template.
They share a staggering amount of their internal architecture, which tells us a lot about their evolutionary past. For a start, both mitochondria and chloroplasts are surrounded by a double membrane, the envelope. But what's more surprising is they replicate themselves independently of when the main cell divides. They just split into two on their own.
They're like these tiny self-governing units living inside the host cell. And they run their own operations with their own gear.
That's it exactly. They both have a circular strand of DNA, totally separate from the nucleus, and they use their own smaller $7 ribosomes.
And that's a huge clue, right?
It's a critical piece of evidence. The presence of that separate DNA in those $7 ribosomes strongly supports the endosymbiotic theory, the idea that these were once independent bacteria swallowed by larger cells billions of years ago.
So they're basically ancient independent factories running inside of you.
Yeah, that's a great way to put it.
It makes the cell floor plan much more of a collaborative commune, not just a single entity. Okay, zooming out from energy, let's look at the assembly line for the cell's workforce, the proteins.
That job belongs entirely to the ribosomes. They are extremely small, only about 25 $5 nanometers across, and they're these highly complex little machines made of RNA and protein.
And their location decides where the final product gets shipped. The cell has this incredibly precise postal system, the secretory pathway.
Exactly. So if a ribosome is just free-floating in the cytoplasm, the protein it makes usually stays inside the cell. But if it's attached to the rough endoplasmic reticulum, the RER, that's different.
That's for export.
It's making proteins for export or for getting embedded into membranes. Those new polypeptide chains move into the RER's network of sacs. They get modified, and then they're pinched off in these little transport vesicles.
And those vesicles are like express mail.
Precisely. They travel and merge to form the Golgi body, which is basically the cell's packaging and distribution center. The Golgi processes them further, sorts them, tags them, and then ships them out of the cell in more vesicles. It's a perfect, unbroken chain of production and delivery.
A marvel of miniaturized logistics.
Now let's focus on the product itself, the
Yeah.
protein. So you have the primary structure, that long chain of amino acids, then it folds. But a lot of critical proteins only become functional when multiple separate chains join together.
All right, we're talking about the highest level of organization, the quaternary structure. How do these huge molecular components find each other, dock perfectly, and then, hold together with all the chaos inside a cell?
That's the big structural question that scientists spent decades on. Take hemoglobin, our classic example. It needs 4 separate polypeptide chains to come together. And what holds them together isn't just one thing. It's a cocktail of 4 specific molecular forces all working at once.
Okay, so what are these 4 pillars of molecular architecture?
You need the stability of hydrogen bonds, the shear strength of strong covalent disulfide bonds, the pull of ionic bonds, and then finally specific interactions between what we call nonpolar R groups.
Hydrophobic interactions.
Hydrophobic interactions, exactly. It takes all 4 of those working perfectly at the nanoscale just to build one functional molecule that can carry oxygen around your body.
And it's amazing to think that if even one of those 4 forces failed, the whole thing would just collapse.
It would.
And we wouldn't know any of this detail without the work of a few brilliant minds decades ago. This is where the history and the science really connect.
Absolutely. The foundational discoveries that unlock these structures, like DNA's double helix in 1953, and then the complex shapes of proteins like myoglobin and hemoglobin, they all relied on a technique called X-ray crystallography.
Right.
And many of the scientists responsible for those Nobel-winning breakthroughs, the Braggs, Sanger, Watson and Crick, Kendrew and Peretz, they were all clustered around the research hubs in Cambridge.
It was such a rapid period of discovery that just fundamentally changed how we see life and what DNA controls ultimately is. Well, everything.
Everything. It's the ultimate code for proteins and enzymes, and since enzymes control all the chemical reactions, DNA is the master controller of every single cell activity.
And that control has to start with a faithful copying process.
Yes. The first major step is transcription, and that happens in the nucleus. Here, the sequence from the DNA sense strand is copied over to messenger RNA, or mRNA. And this whole process needs an enzyme, RNA polymerase, which is the physical worker that moves along the DNA, building that temporary RNA message.
But what happens when that copying process fails? Even a tiny mistake in the code can have massive cascading effects on the final protein.
And that unpredictable change is what we call a mutation. A mutation is just, by definition, an unpredictable change in the DNA nucleotide sequence. Even a single base substitution can result in a polypeptide that doesn't work right.
And we have to remember all the different versions of genes, the allele,
Sure.
they all originally come from mutations.
They do. The classic, most powerful example of this in humans is the HBS allele, the one that causes sickle cell disease. It all started with one tiny change, a single base substitution in the DNA sequence.
And it changes just one amino acid.
It's one. But it transforms the shape of the hemoglobin protein, which then warps the red blood cells into that characteristic sickle shape. A tiny molecular change with massive physiological consequences.
So we've talked about the precise machinery of DNA and proteins, but maybe we should take a step back and appreciate the simple molecule that makes all of this possible.
We need to talk about water.
Let's talk about water. You could argue it's the most important biochemical of all. It makes up 60% of your body mass and 70 to 95% of a cell's mass?
It's the matrix of life, and its incredible properties all come from just one thing: hydrogen bonding. These bonds are weak on their own, but when you get billions of them together,
They give water some almost magical powers.
they really do.
What's one property that you think is the most biologically essential for survival?
I'd have to choose the high latent heat of vaporization. Because those hydrogen bonds hold the molecules together so tightly, it takes a huge amount of energy to break them and turn liquid water into a gas.
Which is biologically very useful.
Incredibly useful. It means evaporative cooling, sweating, or a dog panting is unbelievably efficient. You can lose a massive amount of heat energy with very minimal water loss.
It's the perfect biochemical air conditioner. Keeps our internal environment stable without us risking rapid dehydration.
Then you've got cohesion and surface tension. The high cohesion water molecules sticking to each other, that's what allows water to be pulled in long, unbroken columns from the roots of a tree,
All the way to the top.
all the way to the very top branches of the tallest trees, completely defying gravity.
The idea that a 100-meter tall redwood tree is essentially using molecular sticky tape to pull up thousands of liters of water is just astonishing.
And that same cohesion creates high surface tension. We see it everywhere, but the classic example is the pond skater. It can literally use the water's surface as a solid habitat. It just walks on water.
Without breaking that tension.
It's a macroscopic effect of hydrogen bonds working right at that boundary layer.
That's a perfect link from molecular physics to the real world. Okay, speaking of maintaining balance, let's talk about how whole organisms regulate themselves internally. Let's talk homeostasis.
Homeostasis is all about keeping internal conditions, things like your temperature, your blood pH, your glucose levels, within very narrow limits. And it relies entirely on the principle of negative feedback.
Right, and for an informed listener, we probably don't need the textbook definition, but let's just appreciate the structure. Why is that feedback loop so effective at keeping things stable around a set point?
Because the whole system is designed to constantly reverse change. You have receptors that detect any deviation from that set point. That's the stimulus.
Okay.
Then a central control processes that input and sends instructions out to the effectors. And the crucial part is that the effectors carry out corrective actions that reverse the initial change. It brings the system back towards the set point.
So it's not a one-time fix.
No, it's a constant fluctuation, just always tapping the brakes and the accelerator to stay on the line.
And for a really mind-bending example of this, under extreme stress, we have to look at the black bear during hibernation.
This is one of the greatest feats of biological engineering, I think. For months, these bears don't eat, they don't drink, they don't urinate or defecate. They're just living off stored fat and muscle protein.
But burning protein creates urea waste, which is highly toxic. If they're not urinating, that should kill them. How do they handle that toxic nitrogen?
This is the genius loop. So, to maintain water balance, their kidneys reabsorb virtually all the urine. The urea that would normally be waste gets recycled by special bacteria in their gut.
Recycled.
The bacteria break the urea down into ammonia and carbon dioxide.
Wait, so the bear is fermenting his own waste inside its gut?
In a way, yes. And here's the payoff. That ammonia then combines with glycerol, which is easy to come by because the bear is breaking down massive stores of fat for energy.
And ammonia plus glycerol lets them do what?
Synthesize new amino acids.
Unbelievable. It's a closed-loop metabolic system. They are using waste products from protein breakdown, combining them with fat breakdown products to build new tissue while they sleep.
It's the ultimate example of nutrient recycling and homeostatic control. It ensures that even under extreme starvation and dehydration, the bear's internal chemistry stays stable enough to survive.
That is just next-level efficiency. Okay, let's connect this deep understanding of control to global health and conservation. When we talk about genetics and medicine, we often mean genetic engineering, which is basically the ability to cut and paste DNA.
Yes, the technology is rooted in some very precise molecular tools. We use restriction enzymes, which are like molecular scissors. They cut DNA at specific sites.
And they often make these staggered cuts, right? The sticky ends.
Exactly. They leave these complementary single-stranded ends, which are perfect for joining DNA fragments together. And to move that new DNA into an organism, you need a delivery vehicle, a vector.
Which is where plasmids come in.
That's right. Those small circular bits of DNA you find in bacteria. We can insert a gene we want into the plasmid using those sticky ends, creating what we call recombinant DNA. The classic application, which changed medicine forever, is using GM bacteria to mass-produce human insulin.
So that's technology solving a human problem. But nature has its own brutal, elegant solutions that are driven by strong selection pressures. You mentioned the HBS allele earlier, which causes sickle cell anemia. Yet this really high-risk allele stays at incredibly high frequencies in places where malaria is common.
This is the perfect biological trade-off. It's a classic example of heterozygote advantage. So individuals who are heterozygous, they carry one normal allele, text HBA, and one sickle cell allele. Text HBS, they are significantly protected against severe malaria.
Even though they don't have full-blown sickle cell disease.
Right. So in malaria zones, carrying that high-risk gene actually gives you a huge survival benefit against the dominant killer. The selection pressure for malaria is so intense that it keeps the HBS allele in the population, despite the severe risk to children born with two copies.
It's just proof that genetic diversity isn't just a nice to have.
No, it's the raw material for adaptation and survival against constantly evolving threats.
And when environmental change happens faster than evolution can keep up,
Mhm.
we have to intervene with these massive tech efforts to preserve that diversity. Which brings us to high-tech conservation.
The scale is immense. I mean, think about the Millennium Seed Bank. The goal there is to store seeds from at least 25% of the world's plants by 2025. It's an insurance policy.
So even if a species goes extinct in the wild, we have the genetic blueprint to regrow them,
Exactly. And for animals, we have the frozen zoos,
storing sperm, eggs, embryos.
all in liquid nitrogen at minus 196 degrees Celsius. And this isn't just a few samples. It's a massive global effort to collect and preserve the genetic resources of endangered species for future breeding programs.
So we've covered a spectacular range of ground, from the, 25 automated ribosomes inside a single cell.
To the Nobel-winning structure of DNA.
We saw how the physics of water allows a pond skater to walk, and how these fundamental rules play out in global challenges, whether it's a bear recycling its own urea or the genetic tug-of-war between malaria and sickle cell.
And we've seen that the most robust, the most surprising facts, they often lie in those tiny, detailed mechanisms. The physics of a water column reaching the top of a redwood, the metabolic engineering of a hibernating bear, and the critical role of $7 ribosomes, suggesting the power plants inside your cells are actually ancient life forms.
It has been an absolutely incredible deep dive into the very instruction manual of life.
So we've established that genetic diversity is the essential raw material for survival and adaptation. But the fact that we now need this massive high-tech infrastructure, the Millennium Seed Bank, frozen zoos, huge cryo-storage facilities, to physically store and curate this diversity, it raises an essential question for you to think about. What does this need for human-managed, cryogenically stored biodiversity tell us about the current speed of environmental change? Are we moving beyond being observers of nature and rapidly becoming the essential technological curators of the biological resources that we used to rely entirely on natural evolution to generate?